Welcome to Click for IB Biology. We're in the genetics topic and we're looking at the interaction of alleles for a gene, and the phenomena we're going to look at in this video is that of codominant alleles. There's a blank version of this presentation, a blank PDF down in the description, and you can download that, annotate that with your own notes as we go along. Then at the end, take the 10-question quiz. The answers are there, and check out your understanding, and that will help you to remember and understand the content. So we're looking at codominance, and for our example, I've chosen what is a type of blood group, but it's not the classic ABO blood group. This blood group is called the MN blood group. It's on chromosome 4. There's a gene called HGNC4708, right? And on that gene, it's coding for a structure which is called a glycophorin. So if I just draw a little image of this, there's a red blood cell, and we have proteins sticking through the membrane like so. And on these proteins, they are identified either as type M or type N. And these are actually the alleles of the gene. So at the same gene loci, here and here, we have two versions of the gene. One version, one allele is called the M gene, and the second one is called the N gene. So these will be ultimately be attaching M's and N's to the cell, depending upon the actual full genotype of the individual. So let's look at those individuals. So here's potential individual 1, and at that gene loci we have big M here and big M there. So this implies that the base sequences are identical, that through protein synthesis they would produce the same structure. So the genotype then is expressed as M, big M, like this, and they are indeed, in fact, the blood group is actually simply referred to as MN. So this is actually the genotype, and this is actually the phenotype, like so. And what would that look like at the cellular level would be an erythrocyte, a red blood cell, in which the proteins sticking out through the cell surface like this were all the same and of the M glycophorin A type. Remember, glycophorin's over here. That's the name of the molecule. So there's glycophorin AM and version N. Okay, so let's go down here now, to the second individual, and what we find here is they have the N and the N type. So their genotype is big N, big N. Their blood group phenotype is going to be of being in blood group NN. It's a fairly simple idea. And then when we look at the cell, if we inspect the molecular structure of their glycoproteins in here, they are all of the N type, like so. Now, of course, there's a third genotype, which is here. And this, of course, is going to be the heterozygote. The ones we've looked at here and here, their alleles are identical, so those are homozygotes. Now we look to the heterozygote. So there we had M and N. So M and N would be our genotype. So we've got two different types of allele. So this is the heterozygote. Now in this case of codominance, what happens is that we end up with a third distinct phenotype. And these people are said to be in blood group MN. And if we look at what that looks like, then we have our M proteins present, and there are N proteins as well. And so in this particular example of codominance, we have one gene, we have two alleles, we have created 3 genotypes, and then we end up with 3 distinct phenotypes. And this is an example of codominance, where each of the alleles plays an equal role in the phenotype of the heterozygote. Okay, there's the link to the next phenomena that we're going to look at this level of explaining genetics and molecular biology. There's a chance to subscribe to the channel. That's greatly appreciated. The playlist is there to inform you of all the genetics videos that we have. See you in the next video.